Hi everyone, this is Dr. Hassi. Welcome back to the channel. Today's topic is genes, chromosomes, and gene expression. So when we say gene expression, it includes replication, transcription, translation to the proteins. So we'll start with Mendelian genetics, basically, and then gene idea. As you know, you need to be familiar with the characters, trait, gene, allele, hybridization, monohybrid, dihybrid, QTL, P1 generation, F1, F2 generation, test cross, phenotype, recessive, dominant, epistasis, multiple alleles, right? So basics of genetics. As you know, so the Mendel and his experiments and his two papers he published in 1865 that actually established the major genetics that we know now, and that he's considered the father of genetics. So Mendel used scientific approach to identify two laws of inheritance, and probability laws govern Mendelian inheritance. Inheritance patterns often more complex than predicted by simple Mendelian genetics. Many human traits follow Mendelian patterns, but some don't follow Mendelian patterns. So Mendel used as his model system pea plants, so piece of psilivum, and there were a lot of advantages of pea, including many different varieties naturally exist, many different traits like flower color, different height, different pot size, seed color, seed size, many traits that actually that that was a good choice of model system for him to study. And his experiments, obviously he did major experiments with more than 7,000 something number of plants, several years. He repeated these experiments and did crossing, selecting, crossing, and then he came up with the results actually, and then published them in two papers. And now we know Mendelian genetics. So make sure that you go over a dominant, recessive, what type of dominance, incomplete, codominance, or just dominant, or complete dominant, and then how many genes, is it monohybrid, dihybrid, or polygenic, and then examples for each, and then make sure that you can actually do Punnett square to actually find the is it a 3-to-1 ratio? Is it 9-3-3-1 ratio, 1-to-1 ratio, 1-to-2-to-1 ratio, or what type of ratio actually exists, and to actually solve the genetic problems. So since the next two chapters are also genetics related, so they are all linked to the Mendelian genetics, let's move to next chapter, which is chromosomal theory, or chromosomal basis and chromosomal theory. So here in this chapter, we have Dr. Morgan and his experiments, and then his choice of model organism, which is Drosophila or fruit flies. And so in his work on chromosomal theory and X-linked, especially X-linked genes or Y-linked or wild type mutants, created many mutants of the Drosophila, and then he crossed them with wild type normal phenotypes, and then he looked at next generations, and then he actually, him and his lab came up with the idea of chromosomal theory, and then also now we use this system mapping the genes on specific chromosomes. So Dr. Morgan and his students worked his lab at Columbia. Basically, they show that inheritance has its physical basis on top of what Mendel studied. And then they also found sex-linked genes. They have unique patterns of inheritance. So this was not the case necessarily in plants, because plants don't have sex chromosomes. So the animals like fruit flies, they have XY system. This is pretty similar to human system. And then he came up with this link gene idea, that if two or more than two genes are on same chromosome, they don't necessarily follow this independent Mendelian genetics rule. And then the last, the alteration of chromosome number, structure, and then what kind of genetic disorders that cause, and exceptions to standard Mendelian inheritance. So always remember, chromosomes can be autosomes, 6 chromosomes. In mammals, XX is female, XY is male, but they carry more than just those gender determining genes. They carry also other genes, other important genes. Genes can be linked or unlinked. A lot of Mendel studied genes were not linked, obviously, but today we have linked genes. They only switch if there's a crossover. Maybe that can provide recombination frequency. We can calculate that with a formula. 1% is 1 MEP unit or 1 centimorgan, and we can find MEP location of the genes on the specific chromosomes. Alteration in number and structure of chromosomes, such as deletion, duplication, translocation, and inversions. Make sure that you actually review these. Aneuploidy like trisomy, monosomy, or polyploidy we see in plants like strawberries. And so make sure that you cover that too. And the next chapter is gene to proteins or gene expression, which includes transcription and translation. So this is a pretty cool chapter actually. So we've seen genes, we've seen chromosome, but what about genotype? And what about when actually they translate into the proteins, what happens? So transcription and translation. So this chapter covers how this carried out in prokaryotes, which they don't have nucleus, versus eukaryotes. They have to start a nucleus, leave the nucleus, go the ribosome in cytoplasm. So we go from DNA to RNA to protein, specifically mRNA to protein, DNA to mRNA to protein. You can also replicate DNA to make more DNA. So make sure that, don't forget about that. And then we can actually transcribe it to mRNA, and then we can take that and we can translate mRNA to do proteins, polypeptides. That's also same time phenotype. So we go from genotype to phenotype. So genes specify proteins, again, via transcription and translation. Transcription is the DNA-directed synthesis of RNA. Eukaryotic cells modify RNA after they transcribe it. Translation is RNA-directed synthesis of polypeptide, and also mutations, important. One or few nucleotides can affect protein structure and function that can cause actually mutations. A mutation can be missense mutation, which is single nucleotide change, and that cause amino acid change in amino acid coding. Nonsense mutants, which is changing again amino acid codon, and then frame shift mutants, which can be deletion or some other type of mutation that actually arise from this. So what causes mutations? It could be errors, X-rays, UV, radiation, chemical mutagens, and etc. that can actually cause these mutations, and then that can actually change the nucleotide and can be missense, nonsense, or frame shift type of mutations. Anti means opposite, exo means out, intro means within, poly means many. So always remember these, and then different types of mutations, and then so again, a gene expression. So gene expression and doesn't end here. There is more into the gene expression, but this is actually this chapter just covers onto the protein. Always remember both transcription and translation have initiation, elongation, and translation, and we have this universal genetic code all living organisms share actually with each other. So keep reviewing these chapters, and make sure don't forget the meiosis chapter, because meiosis is heart of this whole genetics. So you make sure that you know 10 steps of meiosis, and then these genes, genetics, genotype, chromosomal theory, phenotypes, proteins. So keep studying and reviewing. Thanks for watching. Please feel free to comment, like, and subscribe. If you like this video, please subscribe to the channel. Happy studying. I'll see you in the next video.